Hi, everyone. My name is Alex Morehead. I'm a upper postdoctoral fellow here at NERSC, also in the cited AI and Learning Systems group. And today I'll be presenting a AI foundation model for 3D molecules and materials called ZAT1. So first off, foundation models. This is a very popular term these days. Models like ChatGPT we're all familiar with. That's an example of a foundation model. It's an AI model that's trained on a vast quantity of data and can be applied across a range of tasks once it's trained. And for science, we're interested in these types of models because we'd like to build, for example, universal algorithms for many different types of scientific data we care about. So in this case, in this work, we're going to focus on chemical systems, so molecules and materials. What would it take to build a foundation for molecules and materials? So the way we look at that is we want to understand how do we represent the data, molecules and materials. So in this work, we're going to argue for breaking down the representation of molecules and materials as atomic systems with 3D points in space and types of atoms, for example, element types attached to them. And we have a foundation model that's trained on these collections of atoms that could be applied across a range of tasks. For example, it could be a universal generative and predictive algorithm. It could generate new molecules and materials. It can also predict properties of existing molecules and materials. It can share information across those different tasks. You could use this in drug discovery, energy research, and many other areas. Now, how would you actually build this if you had a collection of atoms and a bunch of data sets with those atoms in them? There's two main approaches to building foundation models in the AI community these days. Many others, but two main ones are autoregressive models and bidirectional models. So BERT is an example of a bidirectional model, and ChatGPT is an example of an autoregressive model. Autoregressive models, you're just predicting one token after the other, and bidirectional models means you can corrupt some tokens in your, like for example, sentences, and ask the model to predict back missing words in the sentence at any position. So it doesn't assume any kind of ordering when you're doing this bidirectional modeling. And for scientific applications, we're going to argue that that's really important, that we don't want to assume that there's a specific canonical ordering of atoms in these molecular or material systems. So we lean towards bidirectional models in this work. Now, talking about what we've actually developed. So ZAT1 is the first model of its kind. It's an AI foundation model for 3D molecules and materials. It can perform both generation of new molecules and materials and prediction of properties like energies, forces, and so on of existing molecules and materials. It's visualized here in the top right-hand corner. Like most foundation models in the AI literature, it's trained in two stages. The first stage is pre-training, and we'll talk about how that's performed in a little bit. And second is fine-tuning. So the idea is you want to train your model on a vast quantity of data during pre-training, so it sees as much as possible. And you want the pre-training task, what it's actually trying to do, to be as generic as possible so it can learn rich information about your input data. And then when you fine-tune it, you want to adapt it to specific related tasks in your domain, like property prediction. And this is all driven, that pre-training phase I mentioned, it's driven by what's called flow matching, which is this really popular technique in the generative AI literature. Basic idea of flow matching is you have two different probability distributions or data distributions, and you're trying to learn an AI model that will basically map samples from one distribution and push them to the other distribution. So why would you want to do that? This is really convenient for data generation. For example, you could have your first data distribution be something like a Gaussian, something you can readily and quickly sample from. And then the second distribution could be like a real data set, like molecules and materials. So if you want to generate new molecules and materials, you can sample from the Gaussian, use your trained AI model, and it can basically turn that Gaussian sample into a real molecular material. Now, empirically, how does ZAT1 do? So we test it across a range of tasks. First, we're going to talk about the pre-training tasks, which are these generative tasks. How well can we generate small and large molecules, kind of split into two different tasks? For small molecule generation, we're almost at a perfect chemical and validity, chemical and structural validity rate, according to this tool called Postbusters, which is well known in the AI literature. Essentially 99% better than any other method in the AI literature. And we also find something quite interesting, which is that training on both molecules and materials is better than training on just molecules alone. So there's some mutually beneficial information models learning when it sees both types of chemical systems during pre-training. Second, for large molecule generation, we're basically better than any AI model in the literature for this data set called GM drugs, large molecules. We're matching the statistics to the data set. So the data set itself has about a 94% Postbusters validity rate, which is just assessing the chemical and structural validity of its molecules. And we're able to match the statistics when we train on this data set, which is a good sign. It means we're modeling that data distribution faithfully. What's cool, though, is this model can not only generate molecules, but it can also generate materials. So in this slide, I'm showing results for material generation as well. there's this new benchmark called the MatGenBench. We evaluate our ZATA-1 model on. And basically, what we find is that training on both molecules and materials, once again, helps us generate new materials better than training on just materials during pre-training. So what does this mean? It means learning on molecules and materials is helping us do materials discovery better. We're able to generate a higher proportion of stable, unique, and novel materials, which is the main metric we're trying to optimize here, and when we train on both molecules and materials. So that was pre-training. We're basically checking the model's ability to faithfully model different data distributions probabilistically. Now, we've already pre-trained the model, and it's learned some hopefully useful information from this pre-training task of generative modeling. Now let's try to adapt it to predictions of properties. So here we're going to do molecule property prediction. So we're going to freeze most of the weights of our neural network and add a few additional weights dedicated to property prediction. And we want to see how well can it balance property prediction across a range of molecular properties, 12 in this case. We're going to measure the error rate, how close our model's predictions got to the ground truth property in a labeled data set of molecules. And here, we're basically able to improve over a multitask property prediction baseline by 58% on average. And we're also able to do this joint molecule material pre-training to get the best performance possible. So the model's seeing molecules and materials during pre-training. That's actually giving us the best performance during this fine-tuning as well. So this is two different areas where this cross-domain knowledge capture during pre-training is helping us not only for generative modeling, but also related prediction tasks. This is the first time this has been shown in the ML literature, generalized across both generative modeling and prediction, where you get this nice cross-domain benefit. Now, how fast is that? It's basically the fastest model we can find in the literature. This is thanks to a lot of optimizations and the software libraries that we're using for building our neural networks. And we're faster, or this largest version of our model is faster than the smallest version of this well-known baseline in the literature called ADIT. And also when we scale up and try to generate larger molecules, those same scaling trends hold. Basically, the largest version of our model is still faster than the largest version of this baseline ADIT model. So in other words, this is a really good sign. It means that we have very high throughput with our model. Now, because we're building this model on very simple principles in the AI literature, we're using what's called a transformer model. This is visualized here in the bottom left-hand corner. This model makes very few assumptions about the input data that you're representing. And that's nice because it means it's very scalable. It means if you increase the size of the model, you're going to see predictable performance improvements coming from the model. And that's what we see here in the right plot. Basically, we have our training loss, which we want to minimize in the top figure. And in the bottom two figures, we have our validation metrics, which we want to maximize. And as we increase the size of our model, visualized in different shapes and colors, we're getting these statistically significant improvements in performance for all those metrics. So that's telling us that this model, if we train it on a larger and larger data set and kept increasing its size, we'd see performance improvements predictably. That's a really good sign for a foundation model. And with that, I'll go ahead and summarize some of the conclusions of this work. So ZATA-1 is a new state-of-the-art molecule generation foundation model that can be applied across a range of tasks. So it can generate new molecules, materials, predict properties of existing molecules and materials, and the representations it learns are broadly applicable across a range of tasks in molecular and material science. It's also very fast to train and to sample from, to actually use in practice. Its performance improves predictably as you increase its size. But we also want to look at a few new things in future work. We want to see about adding new data with weights. Right now, what are some molecules and materials, but what about proteins and RNA? It's related systems. Could we adapt those to the system as well? And it mutually benefits from those. And second, we want to understand the actual limitations of these bidirectional type of models for scientific applications. Where are they good and where are they worse than maybe autoregressive models? We really want to understand that kind of future work. And with that, I'll wrap up and just thank the funders of this research and the many core contributors and collaborators who made this possible. Thank you so much.